There is a list that infectious disease physicians do not publish. But they know it, the bacteria that antibiotics cannot touch anymore. The fungi spreading through ICUs that no antifungal controls. The organisms that patients check into hospitals with and never check out from. Not because medicine did not try, but because the drugs that were supposed to work simply did not. These are the bacteria doctors are worried about, and the 20 plants on today's list have been studied specifically against them. Not against the easy bacteria, against MRSA, against drug-resistant H. pylori, against Candida auris, against tuberculosis strains that have evolved past every line of pharmaceutical treatment. Starting with a plant almost entirely unknown in Western medicine that has been studied against drug-resistant tuberculosis, a condition for which the options are genuinely running out. And number 5 has a 2018 university study finding its activity against MRSA comparable to vancomycin, the antibiotic that medicine calls the last resort. Number 1. Cape aloe, Aloe ferox, the South African aloe species distinct from the common aloe vera, whose anthraquinone compounds, including aloe imodin and barbaloin, have demonstrated activity against Mycobacterium tuberculosis, including drug-resistant strains in laboratory research. While remaining almost entirely absent from Western herbal medicine awareness, despite significant traditional use across southern Africa, Cape aloe is not the aloe vera sitting on kitchen windowsills. Aloe ferox, the bitter aloe native to South Africa, produces a resinous latex from the leaf base with a distinct compound profile that has attracted tuberculosis research attention for reasons that the common aloe gel does not share. Mycobacterium tuberculosis kills more people globally than almost any other single infectious organism, and the emergence of extensively drug-resistant tuberculosis strains, resistant to both first-line and second-line pharmaceutical antibiotics, represents one of the most serious challenges in infectious disease medicine. Laboratory research has documented that anthraquinone compounds from aloe ferox, particularly aloe imodin and barbaloin, demonstrate inhibitory activity against Mycobacterium tuberculosis strains, including drug-resistant isolates. The mechanism involves disruption of bacterial cell wall synthesis, a particularly important target in mycobacteria, which have unusually complex cell walls that contribute to their resistance to many conventional antibiotics. Traditional healers across the Xhosa, Zulu, and other southern African cultures have used Cape aloe preparations for respiratory infections and chest conditions for generations. The pharmaceutical research community studying drug-resistant tuberculosis has noted this ethnobotanical use and is actively investigating the underlying mechanisms. In Western herbal medicine, Cape aloe is essentially unknown. That is not because the research is absent. It is because the distribution infrastructure for this specific species has not developed in the way it has for its more famous relative. Number 2 has an antimicrobial mechanism, so fundamentally different from how conventional antibiotics work, that the bacteria which have evolved to resist pharmaceutical treatment remain completely vulnerable to it. The compound it produces literally inserts itself into bacterial DNA, a mechanism that no currently available antibiotic uses, and disrupts replication from the inside. Number 2. Cryptolepis sanguinolenta, the West African vine whose alkaloid cryptolepine operates through DNA intercalation, physically inserting itself into the bacterial DNA helix and disrupting replication through a mechanism so structurally different from conventional antibiotics that MRSA strains with full pharmaceutical resistance retain complete vulnerability to it. Cryptolepis sanguinolenta is a woody vine used in traditional medicine across Ghana, Nigeria, and Sierra Leone for generations against malaria, typhoid, and bacterial infections. The alkaloid cryptolepine has attracted serious pharmaceutical research attention specifically because of its DNA intercalation mechanism, a mode of antimicrobial action where the compound physically inserts itself between the base pairs of the bacterial DNA helix, causing frameshift mutations and disrupting replication. This is not how any currently available antibiotic works. Conventional antibiotics target specific bacterial enzymes or cell wall components that bacteria can evolve alternate versions of. DNA intercalation targets the structural geometry of the DNA helix itself, something bacteria cannot easily modify without compromising their own replication. MRSA strains with full resistance to methicillin, vancomycin intermediate resistance, and resistance to multiple antibiotic classes have been tested against cryptolepine and retain significant vulnerability. Clinical research in Ghana has studied cryptolepis preparations for uncomplicated malaria with outcomes comparable to chloroquine in drug-sensitive strains, and with activity against chloroquine-resistant Plasmodium falciparum, where pharmaceutical treatment is failing. The compound that traditional West African medicine identified through centuries of observation is now being studied by pharmaceutical researchers as a structural template for designing the next generation of antibiotics against drug-resistant organisms. Number 3. Black walnut, Euglons nigra, containing juglone, a naphthoquinone compound that kills Candida species resistant to pharmaceutical antifungals through an oxidative stress mechanism distinct from azole antifungals like fluconazole, addressing drug-resistant fungal infections that have become a growing crisis in immunocompromised patients and intensive care units. Candida auris emerged as a global health threat approximately a decade ago and has since spread across intensive care units on multiple continents. It is resistant to all 3 major classes of antifungal drugs in some strains and has mortality rates in hospitalized patients that exceed 50% in some case series. The pharmaceutical response has been limited because Candida auris evolves resistance rapidly and the antifungal pipeline is thin. Black walnut hull contains juglone, a naphthoquinone that generates reactive oxygen species within fungal cells, producing oxidative stress that overwhelms the organism's antioxidant defenses. This mechanism operates through a pathway entirely distinct from azole antifungals like fluconazole, which inhibit ergosterol synthesis in fungal cell membranes. Laboratory research has confirmed juglone activity against multiple Candida species, including fluconazole-resistant strains and Candida auris isolates. The oxidative mechanism also demonstrates activity against several bacterial species and intestinal parasites, making black walnut hull one of the broader spectrum botanical antimicrobials available. Traditional use across Native American and European herbal medicine consistently prescribed black walnut hull for intestinal parasites and fungal conditions, applications that align precisely with the oxidative mechanism that modern research has now characterized. Number 4. Neem, Azadirachta indica, whose nimidin and azadirachtin compounds have demonstrated activity against Mycobacterium tuberculosis, including drug-resistant strains against MRSA through a membrane disruption mechanism, and against multiple bacterial biofilms in a plant that has been the foundation of Ayurvedic medicine for over 4,000 years. Neem is one of the most pharmacologically investigated plants in Ayurvedic medicine, with over 130 bioactive compounds identified from various parts of the tree. The antimicrobial research on neem is extensive and covers a range of drug-resistant organisms. Nimbidine, one of the primary active compounds from neem seed oil, has demonstrated activity against Mycobacterium tuberculosis in laboratory conditions, including strains resistant to isoniazid and rifampicin, the 2 primary first-line tuberculosis antibiotics. The mechanism involves disruption of the mycobacterial cell membrane and interference with bacterial respiration. Against MRSA, neem compounds disrupt cell membrane integrity through mechanisms distinct from beta-lactam antibiotics, meaning MRSA's methicillin resistance mechanism does not confer protection. Neem also demonstrates significant antibiofilm activity against several species, disrupting the protective biofilm matrix that drug-resistant bacteria construct to shield themselves from antibiotic penetration. This antibiofilm activity is clinically significant because biofilm-embedded bacteria are typically 10 to 1,000 times more resistant to antibiotics than their planktonic counterparts. Traditional Ayurvedic medicine uses every part of the neem tree, bark, leaf, seed, oil, and root for different therapeutic applications. The antibacterial applications have been continuously documented in the Ayurvedic literature for over 4,000 years. The research confirming the mechanisms was published in the last few decades. Number 5. Yarrow, Achillea millefolium, whose 2018 University of Thrace study found that yarrow essential oil demonstrated activity against MRSA comparable to vancomycin, the antibiotic that medicine calls the last resort for MRSA infections, through a mechanism involving membrane disruption alongside the hemostatic acolytin compounds that simultaneously prevent the bleeding through which bacterial entry occurs. Vancomycin is the antibiotic that infectious disease physicians reach for when everything else has failed against MRSA. It is called the antibiotic of last resort because when it fails, and vancomycin intermediate and vancomycin-resistant Staphylococcus aureus strains are documented in spreading, the pharmaceutical options become extremely limited. A 2018 study from the University of Thrace in Greece found that yarrow essential oil demonstrated minimum inhibitory concentrations against MRSA comparable to vancomycin in controlled laboratory conditions. The mechanism involves the volatile compounds in yarrow, camphor, borneol, cineol, and various flavonoids, disrupting bacterial cell membrane integrity through multiple simultaneous interactions. The camphor and borneol penetrate the membrane lipid bilayer and disrupt the ion gradients essential for bacterial survival. The flavonoids inhibit bacterial enzyme systems that the membrane disruption leaves exposed. Meanwhile, achillodin, yarrow's hemostatic compound, promotes platelet aggregation that stops bleeding at wound sites, reducing the entry point through which bacteria establish infection. A plant named for Achilles, used to treat wounds of warriors for 2,000 years, has been confirmed in a 2018 university study to demonstrate activity against the bacterium that doctors are most worried about at concentrations comparable to their most powerful antibiotic reserve. Number 6. Grapefruit seed extract, containing polyphenolic compounds including naringenin and limonoids, with documented broad-spectrum activity against E. coli, Salmonella, Staphylococcus, and Candida species. Operating through membrane disruption and bacterial respiratory chain interference, a plant-derived antimicrobial that almost nobody associates with fighting drug-resistant bacteria. Grapefruit seed extract occupies a peculiar position in antimicrobial research. It is widely known in natural health communities, but almost entirely absent from mainstream antimicrobial discussion, despite a research profile that deserves more attention. The polyphenolic compounds in grapefruit seed, particularly naringenin, limonoids, and various flavonoid glycosides, have documented activity against gram-positive and gram-negative bacteria, fungi, and some viruses. The mechanism operates through disruption of bacterial cell membrane integrity and interference with the bacterial respiratory chain, the electron transport pathway that bacteria use to generate energy. Disrupting both membrane integrity and energy production simultaneously creates a bactericidal effect that is difficult for bacteria to develop resistance to because both targets are essential for survival. Research has documented activity against E. coli, including drug-resistant strains, Salmonella, Staphylococcus aureus, including MRSA, Candida species, and several other organisms of clinical concern. The fact that this extract comes from the seed of a fruit available at every grocery store and that its antimicrobial profile against organisms doctors are actively worried about has been documented in peer-reviewed research, makes it one of the more accessible entries on this list. Quick pause. If this list is making you think about having some of these plants available, not just knowing about them, but knowing how to actually prepare and use them, there's a complete practical guide in the first pinned comment below. Step-by-step protocols for growing, preparing, and using the most effective medicinal plants at home. The knowledge in this video becomes real when you have the system to apply it. Link is pinned below. Number 7. Usnea, the lichen growing on trees across temperate forests worldwide, containing usnic acid with documented activity against Mycobacterium tuberculosis, including drug-resistant strains. Abandoned as a pharmaceutical antibiotic candidate, not because it failed efficacy testing, but because its formulation challenges made it commercially less attractive than synthetic alternatives. The story of Usnea and drug-resistant tuberculosis is one of the more instructive examples of how pharmaceutical economics shapes what medicine develops and what it abandons. Usnic acid demonstrated activity against Mycobacterium tuberculosis in mid-20th century research. The activity was confirmed. The development was deprioritized because usnic acid's poor water solubility created formulation challenges that made it less commercially convenient than synthetic compounds that could be produced, patented, and distributed more easily. The antimicrobial activity did not change. The pharmaceutical investment did. Drug-resistant tuberculosis, including multi-drug-resistant and extensively drug-resistant strains, is now one of the most serious infectious disease challenges globally, with the pharmaceutical treatment options genuinely running out in some cases. The compound that demonstrated tuberculosis activity and was set aside for commercial reasons is still growing on trees and forests everywhere. More recent research has confirmed US nucleic acid's activity against MRSA, Streptococcus, and Mycobacterium species, and has identified alcohol-based extraction as an effective solution to the solubility challenge that pharmaceutical developers found inconvenient. Usnea tincture, prepared by extracting the lichen in high-proof alcohol, is used by traditional herbalists for respiratory infections, urinary tract infections, and wound care with a research profile that deserves far more attention than it receives. Number 8. Propolis, the resinous substance honeybees collect and use to sterilize their hives. Containing over 300 identified bioactive compounds with documented activity against MRSA, Candida auris, H. pylori, and multiple drug-resistant organisms, representing the antimicrobial product of 300 million years of evolutionary pressure to maintain colony sterility. The honeybee colony faces a microbial challenge that makes human hospital infection control look straightforward. Tens of thousands of organisms living in close proximity, producing and storing food in warm, humid conditions that are ideal for pathogen growth. The antimicrobial substance bees have evolved to address this challenge. Propolis, collected from tree resins and processed with bee enzymes, has been continuously refined by evolutionary selection for 300 million years. The result is a substance with over 300 identified bioactive compounds, each with individual antimicrobial activity that together produce synergistic coverage against an extraordinary breadth of organisms. Research has documented propolis activity against MRSA through disruption of bacterial membrane integrity and inhibition of cell wall synthesis. Activity against Candida auris, the emerging fungal pathogen spreading through ICUs globally, has been documented in laboratory conditions. H. pylori activity has been studied in clinical contexts. The diversity of the compound profile is precisely what makes resistance development against propolis so structurally challenging. Developing resistance to 300 simultaneous mechanisms would require an organism to undergo simultaneous mutations in hundreds of different genes, which is functionally impossible within clinical timeframes. The 300-compound antimicrobial that evolved to keep a bee colony sterile is available as a tincture, spray, or lozenge at any health food store. Number 9. Pau d'Arco, Tabebuia impetigosa, whose lapachol and beta-lapachone demonstrate activity against Candida species, resistant to pharmaceutical fluconazole through a quinone-mediated oxidative stress mechanism that operates through a completely different pathway than azole antifungals, making it active against exactly the strains that pharmaceutical treatment is failing. Fluconazole resistance in Candida species is a growing clinical problem. The azole antifungals, fluconazole, itraconazole, voriconazole, work by inhibiting the enzyme lanosterol demethylase in the ergosterol synthesis pathway of fungal cells. When Candida species develop mutations in this enzyme or upregulate efflux pumps that remove azoles from the cell, they become resistant to the entire azole class simultaneously. This is the mechanism behind fluconazole-resistant vaginal candidiasis, resistant oral thrush in immunocompromised patients, and resistant systemic Candida infections in hospitalized patients. Pau d'Arco's lapachol and beta-lapachone operate through quinone-mediated oxidative stress, generating reactive oxygen species that overwhelm the fungal cell's antioxidant defenses, a mechanism entirely unrelated to the ergosterol pathway that azole is target. This means that Candida strains that have developed full fluconazole resistance through ergosterol pathway mutations retain complete vulnerability to lapachol's oxidative mechanism. Laboratory research has confirmed Pau d'Arco activity against fluconazole-resistant Candida strains specifically, not just drug-sensitive baseline strains. Indigenous Amazonian use of Pau d'Arco for fungal infections, documented across multiple tribes with no contact with each other, consistently identified the same application that modern antifungal research is now validating against the specific resistant strains that pharmaceutical medicine is struggling to address. Number 10. Oil of oregano, whose carvacrol attacks bacterial cell membranes, disrupts biofilm formation, and blocks quorum sensing simultaneously. 3 mechanisms that no single antibiotic addresses together. With laboratory studies documenting activity against clinical MRSA isolates, collected from actual infected patients rather than laboratory reference strains. The distinction between clinical MRSA isolates and laboratory reference strains matters enormously for antimicrobial research. Laboratory reference strains are standardized bacterial cultures maintained in research settings. They are useful for comparing compounds, but they do not represent the actual diversity and adaptation of bacteria circulating in hospitals and communities. Clinical isolates are bacteria collected from actual infected patients. They carry the full range of resistance mechanisms and adaptations that real-world MRSA has developed. Carvacrol, the primary volatile phenolic compound in high-quality oregano oil, has demonstrated inhibitory activity against clinical MRSA isolates in laboratory conditions, not merely against standardized reference strains. The mechanism involves three simultaneous bacterial targets. First, carvacrol increases cell membrane permeability, disrupting the ion gradients that membrane integrity maintains and allowing cellular contents to leak. Second, it interferes with biofilm formation. MRSA is a particularly aggressive biofilm former, and its biofilm-embedded state is a primary reason it resists antibiotic treatment in clinical settings. Third, it disrupts quorum sensing, the chemical signaling system that bacteria use to coordinate collective virulence behaviors. Targeting membrane integrity, biofilm, and quorum sensing simultaneously through a single compound creates a resistance barrier that single-mechanism antibiotics simply do not pose to adaptable bacteria like MRSA. Number 11. Berberine, from golden seal, barberry, and Oregon grape, demonstrating biofilm disruption in MRSA and Clostridioides difficile at concentrations that do not kill the bacteria directly, meaning it dismantles the protective structure that makes drug-resistant bacteria thousands of times more resistant to antibiotic treatment. Clostridioides difficile, C. diff, is the bacterium responsible for the most common hospital-acquired infection in the United States, causing severe and sometimes fatal colitis in patients whose normal gut bacteria have been depleted by antibiotic treatment. The irony is complete. Antibiotics cause C. diff infections by eliminating the gut bacteria that would normally suppress it. C. diff forms highly resistant spores and produces toxins that damage the colon lining, and recurrent C. diff infection, where the infection returns after treatment, is a significant and growing clinical problem. Berberine has demonstrated activity against C. diff through mechanisms that include direct inhibitory activity against the organism and, more significantly, inhibition of C. diff toxin production. It also disrupts the biofilm that MRSA forms on surfaces and within tissues, the protective matrix that makes biofilm-embedded bacteria between 10 and 1,000 times more resistant to antibiotics than their free-floating counterparts. The biofilm disruption occurs at concentrations below the minimum inhibitory concentration for the bacteria themselves, meaning berberine dismantles the protective structure without needing to be present at levels that kill directly. This sub-inhibitory biofilm disruption activity makes it a potentially valuable combination partner with conventional antibiotics, making drug-resistant biofilm-embedded bacteria accessible to pharmaceutical treatment they would otherwise evade. Number 12. Tea tree, Melaleuca alternifolia, whose terpenen 4-ol is used in certified clinical wound dressings in hospital settings for MRSA-colonized wounds, with no documented resistance development in MRSA strains exposed to therapeutic concentrations. A finding with significant implications given how rapidly MRSA develops resistance to pharmaceutical antibiotics. The absence of documented resistance development is as significant as the antimicrobial activity itself. MRSA acquired its methicillin resistance, the resistance that defines it, through horizontal gene transfer of a modified penicillin-binding protein. It has since accumulated resistance to multiple additional antibiotic classes through various mechanisms. The speed at which MRSA develops resistance to new pharmaceutical antibiotics is part of what makes it such a serious clinical challenge. Terpenen 4-ol, the primary active compound in tea tree oil, disrupts bacterial cell membrane integrity through physical interaction with the membrane lipid bilayer, altering its structure and permeability. This membrane disruption mechanism makes resistance development structurally more challenging because it targets the fundamental physical structure of the membrane rather than specific enzyme targets that bacteria can evolve alternate versions of. Research groups specifically investigating resistance development in MRSA strains exposed to therapeutic concentrations of terpene 4-OL have not documented the emergence of resistance, a finding that contrasts with the rapid resistance development observed with pharmaceutical antibiotics used in the same experimental conditions. The regulatory recognition of this evidence has resulted in medical-grade tea tree oil wound dressings receiving clinical approval in several countries for MRSA colonized wound management. Number 13. Wormwood, Artemisia absinthium. From the Nobel Prize-winning genus with sesquiterpene lactones, demonstrating activity against H. pylori resistant to clarithromycin, the primary antibiotic used in standard H. pylori eradication therapy, through mitochondrial electron transport disruption, that clarithromycin resistance mechanisms do not protect against. Helicobacter pylori infects approximately half the global population and is the primary cause of peptic ulcers and a significant risk factor for gastric cancer. Standard treatment, triple therapy combining 2 antibiotics with a proton pump inhibitor, has eradication rates that have been declining as clarithromycin resistance in H. pylori strains increases globally. In some regions, clarithromycin resistance now exceeds 20% of H. pylori strains, making standard triple therapy fail in a significant proportion of patients. Artemisia species, the genus that produced artemisinin, the Nobel Prize-winning anti-malarial, contain sesquiterpene lactones including absinthin and artabsin that have demonstrated activity against H. pylori in laboratory research. The mechanism involves disruption of the mitochondrial electron transport chain in the target organism, producing an energy crisis that is distinct from the ribosomal mechanism through which clarithromycin acts. Clarithromycin resistance in H. pylori involves mutations in the 23S ribosomal RNA that prevent the antibiotic from binding to its target. Mutations that have no protective effect against the energy disruption that Artemisia compounds produce. A short course of wormwood preparation for H. pylori treatment following failed antibiotic therapy represents an application that the research profile supports, even if it remains outside mainstream clinical guidance. Number 14. Deglycyrrhizinated licorice, DGL, whose flavonoid compounds demonstrate H. pylori eradication rates comparable to standard triple antibiotic therapy in some clinical comparisons. While the related glycyrrhizin compound shows antiviral activity against herpes simplex viruses that have developed resistance to acyclovir, the primary pharmaceutical antiviral treatment. Acyclovir-resistant herpes simplex virus represents a growing problem, particularly in immunocompromised patients. Those with HIV, transplant recipients on immunosuppression, and cancer patients undergoing chemotherapy. In these populations, herpes simplex infections can be severe and even life-threatening, and when the virus develops resistance to acyclovir, which occurs through mutations in the viral thymidine kinase or DNA polymerase, the treatment options narrow significantly. Glycyrrhizin from licorice root demonstrates antiviral activity against herpes simplex viruses, including acyclovir-resistant strains, through mechanisms that include inhibition of viral protein synthesis and disruption of viral envelope formation, mechanisms that do not depend on the thymidine kinase pathway that acyclovir resistance affects. For H. pylori, the flavonoids in DGL, particularly glabridin and liquiritin, have demonstrated antibacterial activity against H. pylori strains, including those resistant to clarithromycin, alongside the additional benefit of stimulating gastric mucus production that protects the stomach lining during infection. Clinical comparisons have found DGL combined with bismuth achieving H. pylori eradication rates comparable to standard triple therapy in some trials, providing an option when antibiotic-based therapy has failed due to resistance. Number 15. Thyme, Thymus vulgaris, whose thymol was the primary pharmaceutical antiseptic before synthetic alternatives replaced it, not because thymol stopped working against resistant organisms, but because synthetic compounds could be patented, and whose documented activity against drug-resistant respiratory pathogens and Candida biofilms remains clinically relevant today. The history of thymol in pharmaceutical medicine is a case study in how patent economics shapes therapeutic development. Thymol was the active ingredient in the original Listerine formulation of 1879. It was used as a pharmaceutical antifungal treatment for hookworm infections before synthetic alternatives were developed. It is still present in several pharmaceutical antiseptic formulations today. Its replacement by synthetic compounds in many applications was not driven by evidence that thymol was less effective against resistant organisms. It was driven by the fact that thymol, as a natural compound, cannot be patented in its original form, while synthetic analogs with similar activity can generate proprietary revenue. Research has continued to confirm thymol's activity against organisms of clinical concern. Studies have documented activity against Streptococcus pneumonia strains, including those with reduced penicillin susceptibility. Activity against Candida species biofilms has been confirmed. The biofilm being the clinically relevant form of Candida in catheter-associated and device-associated infections. Respiratory pathogen activity against organisms involved in community-acquired pneumonia and bronchitis has been documented consistently. The compound that was the foundation of pharmaceutical antiseptics before the patent era remains active against the organisms that the pharmaceutical industry's patented successors are struggling to address. Number 16. Clove, Syzygium aromaticum, whose eugenol disrupts bacterial biofilm formation in MRSA, E. coli, and Candida at concentrations below the minimum inhibitory concentration required to kill the organisms directly, making it a potential combination agent that dismantles resistance infrastructure without the concentration requirements of direct killing. Sub-inhibitory biofilm disruption is a concept with significant clinical implications. When eugenol disrupts biofilm formation in MRSA at concentrations below those required to kill the bacteria directly, it is not acting as an antibiotic in the conventional sense. It is dismantling the protective infrastructure that makes drug-resistant bacteria inaccessible to the antibiotics that are trying to kill them. A clinical application of this concept, combining eugenol-containing preparations with conventional antibiotics to disrupt biofilm and render the biofilm-embedded bacteria accessible to pharmaceutical treatment they would otherwise evade. Laboratory research has demonstrated that eugenol pre-treatment of MRSA biofilms followed by antibiotic treatment produces significantly greater bacterial kill than antibiotic treatment alone. This synergistic interaction between a botanical compound and pharmaceutical antibiotics represents one of the most practically significant research findings in plant antimicrobial research, not using plants instead of antibiotics, but using them to make failing antibiotics work again. Eugenol remains in active clinical use in dentistry as an antiseptic and anesthetic, meaning its safety profile for clinical application is well established. The compound that built ancient spice trade routes is being studied as a tool to restore the effectiveness of the antibiotics that drug-resistant bacteria are defeating. Number 17. Cat's claw, Uncaria tomentosa, whose oxindole alkaloids enhance the immune surveillance mechanisms that identify and destroy organisms resistant to pharmaceutical treatment, including Pseudomonas aeruginosa, the gram-negative pathogen most associated with fatal hospital-acquired infections in immunocompromised patients. Pseudomonas aeruginosa is the gram-negative bacterium that infectious disease physicians find most challenging in severely ill and immunocompromised patients. It is intrinsically resistant to many antibiotics, acquires additional resistance rapidly, forms aggressive biofilms, and exploits immunocompromised states to cause infections with high mortality. The pharmaceutical approach to Pseudomonas relies on the few remaining antibiotic classes that retain activity: carbapenems, colistin, and combinations. But carbapenem-resistant Pseudomonas has spread to become a serious global problem. Cat's claw alkaloids enhance multiple immune mechanisms that are the body's primary defense against Pseudomonas in immunocompromised patients. Phagocytosis enhancement, the process by which immune cells engulf and destroy bacteria, has been confirmed in research on oxindole alkaloids from Uncaria tomentosa. Natural killer cell proliferation and activation has been documented. Macrophage activity enhancement against intracellular pathogens has been studied. Against directly resistant organisms like Pseudomonas that can evade pharmaceutical treatment, enhancing the immune mechanisms that operate independently of antibiotic targets represents a fundamentally different approach. Not trying to kill the bacterium with a compound it has evolved to resist, but enhancing the immune system's own capacity to identify and destroy it through the mechanisms that bacteria cannot easily evolve around. Number 18. Andrographis paniculata, whose andrographolide inhibits NF-κB signaling to reduce the inflammatory tissue damage that drug-resistant infections exploit to establish themselves, alongside direct antimicrobial activity against Streptococcus and Staphylococcus species, and a Swedish randomized controlled trial finding comparable outcomes to antibiotic treatment for uncomplicated sinusitis. Drug-resistant infections exploit inflammation in ways that complicate treatment beyond the direct antimicrobial challenge. When bacteria cause tissue inflammation, the inflammatory response damages tissue and creates conditions. Reduced local blood flow, hypoxic microenvironments, inflammatory debris that actually protect bacteria from immune clearance and reduce antibiotic penetration into infected tissue. Andrografolide inhibits NFKB, the transcription factor that drives the inflammatory cascade, reducing the inflammatory environment that drug-resistant bacteria use to their advantage. This anti-inflammatory activity is separate from and complementary to andrographolide's direct antimicrobial activity against Streptococcus pyogenes, Staphylococcus aureus, and other respiratory pathogens. The Swedish randomized controlled trial that found andrographis preparation comparable to antibiotic treatment for uncomplicated sinusitis is clinically significant because sinusitis is one of the most antibiotic overprescribed conditions in outpatient medicine. The vast majority of cases are viral or self-limiting. Antibiotics are frequently prescribed despite guidelines recommending against it. And this unnecessary antibiotic use is actively contributing to resistance development. A botanical preparation that produces comparable outcomes to antibiotics for this specific indication represents an opportunity to reduce the antibiotic overuse that is driving resistance while still providing effective treatment to patients who need it. Number 19. Olive leaf, Olea europaea, whose oleuropein demonstrates antiviral activity against influenza A and B viruses, including strains resistant to oseltamivir, Tamiflu, through viral envelope protein disruption that operates independently of the neuraminidase inhibition mechanism that Tamiflu resistance affects. Oseltamivir resistance in influenza viruses has been documented and monitored since the widespread use of Tamiflu became standard. Resistance emerges through mutations in the neuraminidase enzyme that Tamiflu targets, primarily the H274Y mutation that reduces oseltamivir binding affinity. When influenza strains with this mutation circulate, Tamiflu treatment produces significantly reduced benefit. Oleuropein, the primary sequiradoid in olive leaves, demonstrates antiviral activity against influenza through a different mechanism. It disrupts the viral envelope proteins that the virus uses for cellular recognition and entry into host cells. This envelope disruption mechanism does not depend on neuraminidase activity and is not affected by the H274Y neuraminidase mutation that confers oseltamivir resistance. Laboratory research has confirmed oleuropein activity against both oseltamivir-sensitive and oseltamivir-resistant influenza strains. The breadth of oleuropein's antiviral activity also extends to herpes simplex viruses, respiratory syncytial virus, and in research models, other enveloped viruses, a broad antiviral profile that reflects the compound's mechanism of disrupting the envelope structures that enveloped viruses share. Traditional Mediterranean use of olive leaves for fever and respiratory infection across thousands of years of consistent empirical observation pointed at exactly the class of activity that modern antiviral research is confirming. Number 20. Garlic, Allium sativum, whose allicin kills bacteria through irreversible inhibition of thiol-containing enzymes essential to bacterial survival, a mechanism that bacteria cannot develop resistance to without eliminating the enzymes they cannot live without, making garlic the antimicrobial that closes this list because it represents the principle underlying all 20. The resistance that defeats pharmaceutical antibiotics was never built to defeat the plants that came before them. Allicin is produced when garlic is crushed, allowing the enzyme alliinase to convert alliin into a lysine in a reaction that begins immediately and reaches maximum yield approximately 10 minutes after crushing. Allicin reacts irreversibly with thiol-containing enzymes in bacterial cells, the sulfhydryl groups of cysteine residues in enzymes essential for metabolism, respiration, and cell wall synthesis. This reaction permanently disables these enzymes. The mechanism cannot be evaded through mutation because the thiol-containing enzymes that lysine disables are not optional. They are essential to basic bacterial metabolism. A bacterium that mutates these enzymes to prevent allicin binding loses the enzymatic function that keeps it alive. This is structurally different from how pharmaceutical antibiotics work. Antibiotics target specific bacterial proteins with specific binding geometries that bacteria can alter through point mutations while preserving the protein's function. Allison's thiol reaction does not depend on binding geometry. It reacts with the fundamental chemistry of the amino acid itself. Clinical research has documented allicin activity against MRSA, H. pylori, including clarithromycin-resistant strains, E. coli, including drug-resistant strains, and multiple organisms that pharmaceutical antibiotics are losing effectiveness against. The 20 plants on this list were not developed in a laboratory in response to the resistance crisis. They were developed by hundreds of millions of years of plant evolution in response to the same microbial challenges that bacteria are now winning against pharmaceutical antibiotics. The bacteria doctors are worried about were never built to defeat these plants. They were built to defeat each other. And the plants were already there. 20 plants. Each one studied specifically against the organisms that drug-resistant infection specialists are most concerned about. Not general antimicrobials tested against easy bacteria, specific activity against MRSA, drug-resistant tuberculosis, fluconazole-resistant Candida, clarithromycin-resistant H. pylori, and the other organisms for which pharmaceutical treatment options are narrowing. This is not an argument against antibiotics. Antibiotics are essential medicine, and serious infections require clinical treatment. This is an argument that the plants preceding antibiotics developed mechanisms against bacteria that pharmaceutical resistance has not yet reached, and that those mechanisms deserve the serious research attention that pharmaceutical economics has not always provided them. Hit the like button if this changed how you think about the resistance problem. Drop a comment and tell me which plant surprised you most. Subscribe and ring the bell. I will see you in the next one.